Fantastic. Thanks very much. And thanks for the opportunity to talk on this topic. Now, several of my ANZICS CTG topics when colleagues, my ANZICS CTG colleagues, when I asked them what was I told them that I was doing a presentation on the TRIPS work of the ANZICS CTG, they said, well, that's going to be a pretty short presentation. But it's given me a topic for a PhD, so there's obviously some work to be done. For those of you who aren't especially familiar with the ANZICS CTG, a brief overview. It's a network of intensive care units around Australia, New Zealand and increasingly other parts of the world, anywhere between 70 and 90 hospital ICUs participating in the design, execution and refinement of large multinational, multi-centre clinical trials to answer some of the key questions in ICU practice today. The trials themselves are uniformly investigator-initiated and nearly all publicly funded and are designed primarily by the methodology centres, which are the ANZICS Research Centre here at Monash, the George Institute for Global Health in Sydney and the Medical Research Institute in New Zealand. In terms of high-impact publications, the ANZICS CTG is one of the most successful clinical trial networks in the world. Since 1998, we've randomised over 50,000 patients in clinical trials. We've enrolled upwards of 30,000 patients in observational studies. We've had over 110 million in research funding and publications of, I think, about 175 peer-reviewed publications, including 15 or 16 in the New England Journal. However, it's fair to say that many of the largest of our randomised controlled trials have not found a difference in the patient-centred outcomes that were expected on the basis of the preclinical and early clinical studies. And I'll touch on this issue a little bit later on. For the purposes of highlighting some of the translation work of the CTG, I'm just going to highlight some of the biggest papers that we've done. And then probably the first and the pivotal one that set the CTG on its path was the SAFE study, which was a multi-centre randomised controlled trial in Australia and New Zealand that enrolled 7,000 patients to receive either normal saline or a colloid intravenous fluid containing albumin for episodes of fluid resuscitation in the intensive care unit. And contrary to the systematic review that had been conducted in the lead up to this study, albumin was not harmful. In fact, there was no difference in mortality between the two groups, except in the subgroup of patients with traumatic brain injury, in whom albumin was found to greatly increase mortality. The second massive fluid study that I want to highlight was the CHEST study, published in 2012. Similar design to the SAFE study, except rather than albumin-containing fluid, we used a different colloid, which was hydroxyethyl starch. And again, contrary to expectation, the more expensive and hitherto thought to be beneficial hydroxyethyl starch fluid was actually found to cause harm to patients in the form of increased episodes of acute kidney injury. And this was subsequently verified by other large international trials conducted by other groups. Back in the ICU in the late 90s, early 2000s, there was a lot of interest in the use of intensive insulin therapy and tight glucose control, which was thought to have a myriad physiological benefits for our ICU patients who are often hyperglycemic. And there were some single-centre trials which seemed to demonstrate really remarkable mortality benefit. And so the CTG conducted the NICE sugar study and definitively showed that, in fact, contrary to expectations, tight glucose control led to greater mortality for ICU patients across subgroups, and this has been confirmed to be because of more episodes of hypoglycemia in the tight glucose control group. And then finally, the last study I want to highlight was the DECRA study, in which patients with severe diffuse traumatic brain injury were randomized either to undergo an operation or to receive optimal medical management for the treatment of intracranial hypertension. And the DECRA study found that although surgery was considered to be the more definitive procedure, and in fact the study showed that the surgery brought down the intracranial pressure faster and more effectively than did medical therapy, the outcomes for patients who underwent surgery was much worse in terms of their functional capacity at 6 months. And that's subsequently been confirmed in a separate trial called Rescue ICP. So all of these studies were interesting for confounding the expectations of the basic scientists of what we understood the physiology to be, and also what some of the single centre and smaller randomised control trials seem to have shown. So if we moved on to our translation efforts, when we talk about fluid resuscitation, to follow on from the SAFE study in 2007, the George Institute did a huge cross-sectional analysis around 340 intensive care units in 54 countries around the world, and found that there was huge variation in practice between centres, between countries, and between specialists. And furthermore, a majority of episodes of fluid resuscitation were undertaken with colloid fluids. So the yellow columns in this graph are colloid fluid resuscitation. The blue is crystalloid, which is normal saline or Hartmann's or Ringer solution. And the column on the column highlighted in red on the far right is all countries combined, and you can see that the tallest column is that for colloid fluid, so most fluid resuscitation episodes were undertaken with a colloid, and this was true to an even greater extent in Australia, and this was despite there being a lack of evidence of benefit for colloids at that time. The George Institute has undertaken this sort of cross-sectional analysis again more recently following the publication of the CHEST trial. And reassuringly, nearly a decade after the publication of the SAFE study and in the years following the publication of the CHEST and other studies, it appears that practice has indeed finally changed such that the preponderance of fluid resuscitation episodes are now undertaken with crystalloid fluids. So what this graph shows is in the y-axis the percentage of all patients receiving fluids. The purple line at the top is the proportion receiving colloid fluids, and the blue line is crystalloids, and the x-axis are the quarters between 2007 and 2014. And there are some of the large trials that have been published in that time. So that's in Australia and New Zealand. Gratifyingly, this pattern of change seems to have extended overseas. So in this work, again by the George Institute and Naomi Hammond, the columns on the left are the results from the 2007 cross-sectional analysis, and on the right from 2014. In the columns closest to me, white is the proportion of crystalloid episodes, and black the proportion of colloids. And you can see that the proportion of fluid resuscitation episodes is, we're using crystalloid fluids, is increasing. And in the columns on the far right, the use of hydroxyethyl starch seems to have reduced massively. And these are good things because colloid fluids are more expensive. They're not beneficial as far as we can tell from the evidence. And in the case of hydroxyethyl starch, they are harmful relative to albumin. So based on the work of the clinical trials group, I think it's fair to say that when it comes to choice of fluid resuscitation, or fluids for used in resuscitation, we have started to generate a significant amount of high quality evidence that warrants translation. Based on Naomi Hammond and the George Institute's work, we have a better appreciation of what practice has been, what it is more recently, and how it's changed over time. I think what has been, and to some extent is still lacking, is an understanding of what are the drivers of that change in that time, and on what basis are clinicians and administrators and policymakers choosing to provide or have available one fluid type over another. And I'll just highlight some work again by the George Institute, and this time by Coleman Taylor, who undertook a very interesting study called a discrete choice experiment. Now, discrete choice experiments would be familiar to anyone who's worked in marketing. I haven't, but I'll do my best to explain it. Basically, in a discrete choice experiment, a consumer, and usually somebody who might be going to buy something, for example, a car, is offered two choices, car A or car B. The characteristics of car A and car B are provided, and they might be the price, the maximum speed, the colour, and the consumer or the respondent to the survey is asked to pick which car. And then those characteristics are varied in a planned statistical way, so that at the end of the analysis, the investigator can see which of the characteristics that consumers value most. And using this methodology, Coleman and his colleagues tried to unpack why some fluids were chosen over others for use in fluid resuscitation. And among doctors and nurses, this study was repeated among different craft groups, and the results varied considerably according to primarily safety, patient types. So doctors were more concerned about which patient groups they were giving which different types of fluids to, the content of the fluids in terms of its salt and colloid content, and other characteristics. So this is some very interesting preliminary work that has the potential to be applied across different interventions that we might wish to unpack. Talking about glycemic control practice for a minute, Maya Kalkinen and Ronaldo Bellomo and Neil Orford looked at the application of tight glucose control in Australian and New Zealand intensive care units in the months before publication and after publication of the NICE sugar trial. And what this graph shows is the blue line is the mean blood sugar on day one of someone's ICU admission. And I'm reassured or I'm assured, I should say, by the text of this publication, that in the second half of that graph, the blue line goes up, indicating that blood sugars were not so tightly controlled in the period after publication of Nice Sugar. And they concluded that on that basis, the Nice Sugar results were being incorporated into ICU practice. And they had, with that, some nice graphs showing that mortality in association with that had gone down. There's a lot of room to unpick these results, so I wouldn't hang your hat on the fact that mortality went down in ICUs based on this single intervention. And I think more robustly and more concerningly, this article from the JAMA Internal Medicine in 2015 looked at the uptake of tight glucose control across intensive care units in the United States, and found that there was fairly consistent implementation of tight glucose control in the decade following the original single centre publication, and then only a slight decrease in the rate of uptake of tight glucose control after Nice Sugar, which I'll just remind you showed that tight glucose control killed people. So that's a bit unfortunate. When we talk about the practice of management of severe TBI patients, Ben Beck here in Belinda's group and used the Victorian State Trauma Registry to look at outcomes following severe TBI across a decade, which included the publication of the DECRA trial, and there's really been no change in either mortality or the functional outcomes of patients with severe TBI in the Victorian jurisdiction. And I'm currently looking at whether our practice has changed in that time as well. And I hope to have those results available pretty soon. So if I can just present a scorecard for the CTG on our translation efforts thus far, I think we've done a reasonable job of identifying and answering some of the key questions in practice in intensive care internationally at the moment. I'll give us 2 out of 3 stars for measuring current practice, but for understanding the barriers and enablers to implementation, I think we only get 1 star. And then for improving practice, I didn't want to actually give a score, knowing that my colleagues might hear about this. So if I can just speculate as to what some of the barriers to implementation might be that are specific to ICU and to the CTG. The first would be that there are actually there is a general poor evidence base for most of the practices that we undertake in intensive care. A lot of it is based on expert opinion, based on the opinion of the clinician at the end of the bed, and based on loose understandings of what the physiological benefit might be to a patient, rather than there being any patient-centered endpoint. And Steve Webb's got some interesting data from Royal Perth looking at the quality of evidence for patients, for consecutive patients for the treatments that they've received over a short period of time. Furthermore, there's a lack of trials, even among the high-quality studies that we have done, that have actually shown an outcome difference that would prompt active implementation by clinicians. So I think that there is a perceived lack of relative advantage of one treatment over another, so why bother? And that might come back to Coleman's work looking at the different characteristics of different treatments, and if we could advertise and increase awareness of costs of one therapy over another, that might address that specific barrier. Just to highlight those 4 trials again as examples of this, the results of our trials have often gone against the expectations that we had when the trials were started, be it because their surrogate endpoints were improved by the trial intervention. In the case of the DECRA study, the intracranial pressure came down, but the patient's outcomes were worse with surgery. This has meant that the interventions that are available at the end of these trials are often incompatible with current practice or with the received wisdom of the day. And finally, I would say that among the CTG leadership and also the CTG community more generally, there is a very strong recognition that high quality research must be undertaken by clinical trial networks in order to be broadly generalisable. But the process of translation has been left to individuals and to individual sites. And there have been occasional attempts to network this, but there's been a few false starts as well. So if I wanted to highlight some of the possible enablers of implementation specific to the CTG, I think that the ANZICS is a fantastic institution. The CTG is all the infrastructure that is necessary to promote translation of research into practice. And furthermore, ANZICS CTG, the ANZICS also has an affiliated registry called the Centre for Outcome and Resource Evaluation, which is actually 4 different registries collecting enormous amounts of data on individual patients and units about outcomes and resource availability. And I think that there should be greater linkage between these 2 branches of the ANZICS, the Intensive Care Society. And last but by no means least, if you've ever spent any time with intensive care clinicians, you'll know that they're hooked on their phones. Social media has got a bit of a bad rap lately, but it's certainly very popular among the intensive care community, and there is a substantial amount of data to support the use of social media in promoting research findings and for implementation as well. So that's the end of my 10 minutes. I'd happily take any questions.